All right, I have the privilege of talking to our second panel today on therapeutics in or very near clinical trials, which is amazingly exciting because when I started 4.5 years ago, I'm Barbara Tate, by the way, Chief Scientific Officer, one panel was enough. We had enough space for all the things that were in clinical trials, and we don't anymore. So that is very exciting. And I have another really geeky scientific excitement that happened today. The FARA and our funding partners fund a group of scientists at the Broad Institute at MIT and Harvard. They call themselves the Friedreich Ataxia Accelerator. And one of those scientists, Gary Rufkin, was awarded the Nobel Prize today. The Nobel Prize in Physiology or Medicine. We funded him before he was a Nobel laureate. All right, so our panelists today are going to talk about, as Jen said, the programs that are more closer to the gene. Not all the way at some points, but closer to that genetic change that causes Friedreich's ataxia. I'm just going to go down the line. I made them sit in order. Rusty Clayton is here from Larimar Therapeutics. Sean Jeffries is here from Design Therapeutics. Franca Angeli is from Lexeo. We have Niyama. Did I say it right? Nave. Yeah. Savage is from Cornell. And then Brian McGill is from Astellas. So maybe we go just skip the next 2 slides. Oh, I have to do it. Jen went over these. It's really, telling you again the molecular basis of the disease and how there are different places where we can intervene to see therapeutic benefit. But I think we can get that same message from the pipeline slide. And we were saying we should have put a scan trot on here so that you can find this easily. It's downloadable. We do update it very often, but it gives you sort of the whole scheme. If you go across the top, you see that we go all the way from idea to approval. And we have something in approval, which is awesome. And then down the side, we talk about how these may affect the disease. And we talked at the earlier panel about things that are in the red. There are sort of legacy programs. There are the historic programs. They're the things that people started on, really understanding that mitochondria were key to the disease and that there were druggable targets there. But as new technology has come into the development of therapeutics, that technology has been applied to FA. And that's the excitement we're going to talk about with this panel. The most cutting edge technologies, they're early, as you see in the pipeline. They're still in the early stages, but they are being applied to FA, and that means that the opportunities for really being able to address underlying features of the disease is possible. So I'm going to start with nomlabofusp. Still didn't say it right. Rusty's been trying to teach me how to say that for a couple of panels now. But this is the Larimar program. If you look back on the pipeline, you'll see the Larimar program is the one, stands out in one approach, which is frataxin replacement. So this is physically giving the protein to cells, the protein that's absent, as a consequence of the GA repeats and the silencing of the gene. This program ambitiously is delivering that frataxin to the body. And we know that frataxin undergoes changes, maturation, to have the protein that's functional. But what Larimar has done to make it possible to deliver that protein is added a little bit of peptide. You see there the cell-penetrating peptide to allow this protein to cross over the cell membrane, actually get into the cell, and then have a function at the level of mitochondria. It was no small task. And in order to make sure that, in fact, these changes were happening, Larimar has committed to looking at changes in the body tissues to confirm that there are increases in frataxin levels in people that have received the drug. So you see here, that's got to be a change from baseline. Since everybody has slightly different frataxin levels, Larimar brought people in, took a skin sample, measured their frataxin levels, and then after 14 or 28 days on drug, they looked again to see if the frataxin levels had changed, had gone up. And you'll see there are two doses represented here. The 25 milligram cohort, we've used the word cohort a few times today, people all getting the same treatment, 25 milligrams, or the 50 milligram. Placebo are people who did not receive the frataxin peptide. So you see two representations of that change from baseline. The one on the left is spatially compromised, so is the mean, so the groups together, but then you actually can see the individual patients as well. And it shows you how they're changing individually over time. And I really like this representation. This is using the 14-day data. And I think it's just a really nice way to see that people in the study from their baseline to 14 days moved through a continuum of measures of frataxin. So those that are low, very low relative to healthy controls, the frataxin level being less than 12.5%, up to those that are 35, 37%. And you see that people started at baseline, and then they move over to they move down at 14 days over to a cohort, a group of people with a higher level of frataxin. And that happened whether it was the 25 mg group or the 50 mg group. There's an open label study that people who had been in that initial study were allowed to enroll in. The open label study is the 25 milligram dose that's given daily subcutaneous injections. And as is true of other open label studies, the key here is to look at long-term safety and viability. Also, though, the efficacy that we can see over time. So that's both by tissue frataxin levels, data we've already presented, but also changes in clinical measures that would indicate that the frataxin that's being delivered is functionally important. And then there is also, excitingly, more trials planned, including pediatric trials. So the open label is OLE, but then the middle panel is plans to initiate a PK study in children before the end of the year. Yes, exciting. And in very young children in 2025. And again, as we've all been hopeful for, we also want to see these drugs available globally. And so there's plans by Larimar to start global studies in 2025. Now, Rusty, a few questions for you before we go on. Can you tell us more about the open label study? Like, can you expand a little bit on what was accomplished during that open label study and if there are any updates?
Would love to do that, Barbara. And before I do that, though, I want to thank you and Jen and Ron and FARA for having this event. I've been there in Baton Rouge and Lisle and Tacoma, and this is the last one this year, and it's just such a joy for me to attend this. And so thank you to FARA for having this event. And I think it's really helpful for the family as well. And then thank you to all the patients that have participated in any of the studies, including the natural history study. It's very important for the folks up here to have those natural history data because it really helps us, figure out what we need to do for the clinical study. So thank you all for that. So with regard to the open label extension, we had our first subjects enroll, our participants, I should say, enroll in March. And those folks are still chugging along. And so they're now, I guess, in their 7th month now. And we've had some other folks come in. And so we have some folks that are 30 days in, 60 days in, 90 days in, and all the way up to close to 200 days in the study. From that standpoint. So we're continuing to enroll. In August, so just a couple months ago, we had some different pieces of information come in, and we decided that we were going to kind of put a pause on anybody who was new in the study until we finished a protocol amendment. And the protocol amendment is we took a look at this information and said, how can we, is there any way we can make the trial even safer? Is there any way we can improve the patient experience? Is there any way that we can enhance, our learnings from that? And so we're in the process of finalizing that amendment. I can tell you the amendment's been sent in to FDA. It's now submitted to the IRB. And the folks that have been dosing continue to dose. The folks that have yet to start day one, we've asked them to hold off for a little bit, but very soon we'll be starting those day one doses, and we'll expect to increase enrollment again through the end of the year. So we'll be chugging along again, with that.
And just to clarify, when you do an amendment to a protocol in an open study, then there has to be a regulatory input on that amendment, correct? And there's some time gap that creates.
Well, right. So there's two things that has to happen. First, anytime you amend a protocol under an IND, an investigation of a new drug application, the FDA has to see that amendment. Now, because of the situation that Larimar is in, we can submit the amendment and go, right? So it's as soon as the FDA receives that, we can act on it. But then there's a second step, because every institutional review board at every place also has to receive that amendment. And because of the nature of the amendment, we also had to change the informed consent forms and some other forms and that sort of thing. The IRB has to look at all of that before we can proceed under that amendment. And we thought that all of the elements of this amendment were so important that we didn't want to have any more day one starts until we had that amendment in place.
That's very helpful. Thank you for that. So then you've got 2 other studies that we have up here. Can you tell us a little bit more about those studies in children and outside of the US? And how can people learn more about that? Where is there a place where they can go to learn more about the possibility of being involved in these studies?
Great question. Thank you. And I'm very excited to talk about the pediatric. This has been a long time coming for us. And as a pediatrician, I'm very excited to now open the program to people under the age of 18. So we will be starting that study in approximately 4 to 6 weeks. It'll be at a single site. The site is called Uncommon Cures, which is located in Chevy Chase, Maryland. No relation to the actor. And we chose Uncommon Cures because they're one of the clinical research units that specializes in taking care of people with rare diseases. And we're very excited to work with those folks. So we're in the process of getting all of that together. In terms of finding out more information, let me tell you a little bit that we're going to be starting out with the older children. So 17 to 12 years of age will be enrolled in the first cohort. It'll be a 7-day study. So there'll be 7 doses. That'll be preceded by a screening period of roughly 30 to 35 days. They'll get dosed for 7 days, and then there'll be a follow-up period. Anybody that study and continues to be eligible will be able to go into the open label extension study like the adults previously. And then once we get the data from the adolescents, then we're going to move into the lower age group, and that'll be sometime next year in 2025. And so we're very excited to get that study kicked off. In terms of where you can find information, I was actually over at the table there looking at some of our patient recruitment materials that is going to go in front of the IRB, and then they'll be out. And among other venues, we're going to work closely with FARA to make sure that FARA is able to get the word out as quickly as possible. So if I were to say any one source, I would say go to the FARA website and keep your ears open for anything that FARA would put out.
So we've talked about phase 1 data, PK data, and safety data. We've talked about the phase 2 data. I presented some of those data, but there's a phase 3 data or phase 3 study as well. Can you tell us a little bit about that? And I'm going to throw in, I love just the geeky part of this. Explain a BLA versus an NDA.
Okay, so an NDA that's short for new drug application. And so there are things called, that we call small molecules. And the small molecules are relatively small. They're like your everyday drugs, like penicillin, for example, would be a small molecule. A BLA is a biologic license application. And so anything that's a biologic could be, for example, a gene, or it could be a protein. In our case, it's a protein. And so it falls under that biologic license application. The processes are very similar. And in fact, we are, even though we're a biologic, so to speak, because of our protein in the FDA, we're actually under CDER, the Center for Drug Evaluation and Research, rather than CBER, the Center for Biologic Evaluation and Research. So it gets very confusing very quickly, and I apologize for that. But it's basically because we're a protein, it's a BLA.
That's great. And the phase 3 study?
Right. So we are looking at starting our phase 3 study about mid next year. It'll be a global study. So the US will certainly be one of the countries, Canada. We're also looking at countries such as Australia, the UK, Europe. We're also considering some other territories as well. We haven't made any final decisions yet there. And we expect that we'll be enrolling folks down to age 2 years of age in that study, and all the way up to, I think. So Ron and I have been partners in crime ever since the workshop in July, was it, with FDA? Yeah, we're trying to move the needle in terms of getting children into studies earlier, and Ron's really done a lot of work, trying to move that forward. Anyway, so yeah, down to age 2, I think the upper limit we're looking at maybe 40. The full inclusion exclusion criteria is still being worked out. We're having discussions with the regulatory agencies, but we're moving forward, and we're hoping, and I think I talked about this when I was in Tacoma. Right now, our drug product is a frozen liquid, and it's not really convenient. But we're working on a lyophilized formulation, and we hope that the lyophilized formulation will be available soon, and that's the formulation we're going to use in our global phase 3 study.
That's very exciting. Thank you so much, Rusty, and thank you for being at all of these. It's always great to see you.
It's my pleasure.
So I'm going to move back to the pipeline slide, and we're going to move down the pipeline now. We're going to talk about a small molecule program, the design program, which is a small molecule that's been given the name of gene tac, whose mechanism of action is to increase the production of your own frataxin levels. So we have here the genetic cause of the disease, which is that GAA repeat. Everybody has GAA repeats. You see in the healthy individual, there are GAA repeats. But in a patient with frataxin, those repeats have been expanded considerably into the hundreds or maybe even into the thousands. And when that happens, much, much less messenger RNA and much, much less protein is made, as shown on the slide. So what a gene tac does in the original molecule was DT216. Keep that in mind. I was going to ask Sean to explain a little bit about where we are now. Is that molecule, that small molecule, is able to bind to the area of the GA repeats and jump over those repeats, that silencing mechanism, and allow the engines that drive messenger RNA production normally in the body to occur. And when there's messenger RNA, then there's protein. So this sort of bypasses that block on the gene and allows normal levels of frataxin to be made. Again, I think this is so great that our partners, our sponsors, are helping us develop not just therapeutics, but things that enable the development of therapeutics. And one of the things that design did was to get all of you, many of you, to come in and give muscle biopsies so that they could assess yet another tissue to figure out what were the frataxin levels in that tissue prior to treatment, and what do they look like after treatment. And these are new data. These are data we did not have before. We didn't understand that muscle could reflect the disease. And there is a difference between a control or somebody that doesn't have those repeats, a carrier who has one gene with repeats and one without, and someone with FA who has two genes with repeats. This is new data, and this is really enabling for the field that we can get an accessible tissue. It's very hard to sample your brain, and hard to, but not impossible to sample your heart, but we can sample muscle. So I know this was a big undertaking, both for design and for the community, but it gives us another tool, and that's great. So what did they find? The drug product, remember, a small molecule, as Rusty said, small molecule comes in and out of the body, and we know a lot about that. We've been doing those kind of drugs for years. You see that the small molecule has an exposure in the body. So we're looking at DT-216 from this first clinical trial, and patients that received 100, 200, or 300 milligrams of the drug. The concentration of the drug goes up, and then over time it drops. And that's what we expect, that the drug would be cleared. If we look at the muscle biopsy data, it also indicates that when the drug was on board, the amount of frataxin, this is still
Still drug levels.
This is still drug levels. Okay. So in the blood or in the muscle, I'm already giving away the punchline, increased. And then we look at muscle levels of messenger RNA, and they also increased. So we had the drug in the right place. It went into the muscle and dosed responsibly. This is really important because it means that the drug is behaving as we would predict. More drug, more change in messenger RNA in the muscle. And you can see that again by means on one side of the slide or by the individual patients that participated in the study. So really exciting. And I know that design is very grateful for the community participation, especially those who went in for a biopsy that didn't get the drug, that were just there to give us those baseline data. But maybe, Sean, you can tell us a little bit about, so 216 is not being developed now.
So that's not quite right. So 216 is actually still in development. We ran a phase 1 clinical trial last year. And in that clinical trial, the way that we delivered the drug, the formulation of the drug, was causing what are called injection site reactions. So these are irritations at the site of injection. The drug was delivered by IV injection. And this really, prevented us from dosing more frequently or for a long period of time, which would be required in a lifelong disease like FA. So we went back and found a set of what are called excipients that we can use to solubilize the drug DT-216 in a way that is gentler on the injection site reactions. And we've seen that we can eliminate these injection site reactions so that it's, there's no irritation at the site of injection. And excitingly, we've also been able to show that now we can not just deliver the molecule by IV injection, but also by sub-Q injection, which can be much more convenient for patients, especially for a long-term therapeutic. So we're bringing both of those routes, both IV and sub-Q, back into the clinic with DT-216 to basically show that the short improvement in frataxin expression that we saw in the study last year can be extended to be able to allow patients to, from their own genome, produce more and more frataxin in an effort to hopefully resolve some of the symptoms.
Right. So 2 clarifying points. So the product is still the same. So DT216, but because the formulation's been changed, it feels like you had to step back and do some of the work again. And when you're saying the two routes of administration, IV or sub-Q, both seem to work, that's actually animal data.
That's
correct?
right.
Yeah.
That's right. So we haven't yet gone back into the clinic. What's exciting about this new formulation is that we're able to achieve much higher and more durable exposure of the DT216 molecule, which hopefully will extend that increase in frataxin expression that we saw in FA patients last year. So we're optimistic that we can continue to both increase and extend the increase in frataxin expression that we saw with the same molecule. So it's the same drug molecule that's being delivered, just delivered in a different way.
So by addressing the formulation, you've gotten an improvement in tolerability or safety, but also in the actual action of the drug.
That's right. Yeah.
That's great. So people did participate in the first trial with DT216 in the first formulation. Will there be an opportunity for them to participate in the new formulation or in another study going down the road?
I mean, we're so grateful for all the patients that participated both in the biopsy study, but also in the both SAD and MAD clinical study. And of course, we have those people top of mind. We have not, finalized our submission to regulatory authorities, and we don't have a formal protocol. But of course, we would like to incorporate them into the development program as soon as possible.
That's awesome. Thank you, Sean. All right, we're moving down the pipeline as well. So now we're getting into the purple area, which is gene replacement. So we talk about gene therapies in a broader sense, and we've had some educational materials on this. You can either affect the gene that's currently in the body, or you can actually give a new gene. And giving a new gene, we call gene replacement. So the next trials, next three that we're going to talk about are all gene replacement trials. And the first one is Lexeo. And there's some really nice material in here that tells us a lot about why. Why did Lexeo choose to do a gene therapy for the heart? And part of that is really driven by understanding that the disease, while it manifests as a neurological disease, this underlying change in the heart is really impactful. It may be part of the symptomology, for instance, the fatigue. It certainly, as we'll see on the next slide, is part of the reason that people do not live a long lifetime. What happens in the heart is in the normal heart, we have the chambers of the heart, and actually Kim went over this earlier today, are flexible, are able to pump the blood. But as the heart responds to the lack of frataxin, the muscles thicken, especially in the ventricles. And when they thicken, the heart doesn't pump as well. And that's called hypertrophy. So that's the range of the disease. As I said, and as Kim said earlier, this is a really impactful aspect of the disease because this is what leads to shortened lifespan. And just the symptomology of it, we know that it can contribute to certainly the irregular heartbeats that Kim talked about, the arrhythmias. It can cause scarring of the heart, fibrosis. And it may be that some of this also affects the ability to do what you want to do. All gene therapies have to be delivered by some mechanism. And the most common is using the proteins that surround a virus to be able to deliver the gene to the cells. Viruses are really good at getting into cells. They usually carry their own genes, their own genetic machinery because they want to reproduce themselves, but we're able to take out all of the virus genes and actually put the gene that we want to deliver. In this case, LX2006. This is delivered by an IV infusion, and that gene then can go to the cells. insert itself into the cells as a working copy, then be transcribed, produce the protein, and the protein, being the normal protein, will find its way to the mitochondria. The Sunrise study, and a little more geeky stuff about drug development here, is called phase 1, phase 2. And I'll explain a little bit about why that is. But this study is for cardiomyopathy and FA. It's for adults. That's not unusual. This is a very cutting edge technology, a very cutting edge approach. And regulatory agencies tend to be a little bit conservative. So starting in adults is not unusual. Participants are able to take Skyclarys, which is great news. Again, you receive an IV infusion or an IV injection. And the first things that we're going to measure, as we do in every phase 1 study, is safety. We're going to look at safety. Now, because you're actually getting an active product and you are a patient with the disease, we will also look at frataxin level changes. And we will also look at changes in the heart. That's what makes it phase 1 and phase 2. We're doing safety, which we do with every therapeutic, but in this case, we're also looking at efficacy markers. It's a long study, duration of 1 year. And as we're seeing with all gene therapy studies, there's a very long follow-on time, another 4 years of follow-on. So in this case, in the Sunrise trial, people were asked to allow a tiny bit of the heart to be biopsied. And that was important because that is the tissue that we hope to be treating. And you had to have a biopsy before treatment and then a biopsy after treatment in order to show the change. So you see the photomicrographs here, the actual histology of the heart of the biopsy, showing you before treatment, there's very little frataxin expression. But 3 months after treatment, when we took another biopsy, you can see the brown staining is frataxin. So there was delivery of the gene. The gene did cause the expression of the protein, and we see it in the right place. So that was very exciting. And thank you all for volunteering to have a piece of your heart biopsied. But it was really important, I think, for this study to have that biomarker to show that there really was the right delivery and the right change in the heart. So there are actually 2 trials underway with this same product, the same gene therapy. One is the Lexeo trial, phase 1 to Sunrise, and one is at Weill Cornell. And that is just a safety study. There is no biopsy in that study, so it's just looking at safety. In both trials, we have the data now from both trials. There's been very well toleration, it's been tolerated and very good safety. So that's great news. And there are some indicators that there could be changes in the heart that are also significant. And the Sunrise trial continues on. There's a table here from Lexeo today. So please do visit the Lexeo table and get more information about the trial. As Rusty said, you can always go on the FARA website. It's very easy to find open trials and to find information about the trial. So either Lexeo or FARA would be happy to give you more information about this. I'm going to stop here and ask a few questions of Franca from Lexeo, and then we'll ask a few questions about the Weill Cornell study as well. So we've noticed that there's a recruitment notice for the Sunrise trial. And one question that came from our community is, so you're aiming to treat the cardiomyopathy. Do you believe that there could be benefit to other tissues?
So first of all, thank you so much. It's a pleasure to be here, and thank you for the opportunity to talk to everybody. We are collecting measures across all tissues to make sure it all functions. And we're going to make sure to report the data. The capsid, the mechanism that we are using to deliver, it is has more affinity to the heart. We know that not only the heart is receiving the gene, the new genes, therefore it could be some benefits. We can only confirm that when we have the data. Yeah, we're only going to be able to demonstrate that when we have all the data collected, but we are collecting measures to look at neurological improvements as well.
Okay, so just to recap, because it was a little difficult to hear you. So your aim, the delivery mechanism that you're using should prioritize delivery to the heart, but that doesn't mean it wouldn't get to other tissues. And you're actually doing other assessments in addition to those that I described to see if there's an impact on other symptomology of FA.
Correct. Correct.
That's great. So once you finish this study, what happens next? So we call this a phase 1, phase 2. What happens? Maybe share somebody else.
Yeah.
It seems that mic has been difficult all day. What happens next? What's the next thing in the development program?
Okay. Can you hear me now?
Much better.
Better. Okay. I am a very soft-spoken person. I apologize for that. So we are very pleased to let everybody know that we are now close for screening for our trial, and we hopefully very soon to end our enrollment in this study. We hopefully going to be able to open, if safety continues to be in favor, next year, our next stage, which will be a registrational effort. Therefore, we are looking forward to collaborate, and hopefully we're going to be presenting the full data set for this study in the next meeting.
That's awesome. All right, thank you. I'm going to just move on to the Wild Cornell study. There are many things that are similar again about this study. We'll just show you a few slides that replicate what Lexeo is doing, but there are some differences as well. And one of those is that this really is just assessing safety. We're not assessing efficacy because there are no biopsies in this study. So far, there are 8 people that have been dosed, and it's again been very safe and well tolerated, no serious adverse events. Preliminary assessments do seem to indicate there may be changes in the heart. And as Kim talked about earlier, there are blood markers, troponin, which could also be going in the right direction. But as Kim also warned you, it's quite a complicated marker. I think one of the things that we're most interested in hearing from Neva is that it's a little bit confusing, having these 2 studies. So understanding that the Wild Cornell study is actually supported by NIH, by an NIH grant. Although it's the same product, there's differences in the governance and oversight. What has happened, though, is that Wild Cornell and Lexeo have agreed to share the data, to put all the data together for regulatory review, which actually allows everything to be accelerated. So the 2 independent trials are running, but the data comes together, and that gives us a larger data set to assess both safety and potentially efficacy. So I think one of the most important slides then for Neva is this. If you want to participate in the trial, there are still slots open. So 2 more slots at the mid dose and 5 slots for the high dose. This would require travel to New York City, but there is travel accommodations and support if you want to be involved in the trial. I think we will share or have shared the contact information. Is there anything else you'd like to add?
No, I mean, thank you for that overview. But yeah, I think we're happy to be here, and we're still recruiting. So if you are interested, please reach out to myself or Maddie. Maddie's down there as well. So we're a small team, but yeah, we're excited to just be here and meet the community. So please ask us any questions or just come and say hi.
And thank you, Lexeo and Wild Cornell, for coming to the agreement to use the data together. It's certainly really does help the community get to answers faster. All right, our last program is also a gene therapy program. Astellas has also started a cardiac study. This is again a gene therapy for cardiomyopathy. Some of these slides again are great just for general information to understand the heart disease and FA a little bit better. We've talked about hypertrophic cardiomyopathy, the thickening of the walls of the heart, which can make it become stiff and not pump as well. Then Kim also talked about the arrhythmias that can be a consequence of low frataxin levels. And eventually, after the thickening, the heart can actually become thinner. And that's a dilated cardiomyopathy. That's a very serious condition. So we really don't want anybody to get to that state. And hopefully these therapeutics will arrest the changes in the heart and restore some of the heart function so that nobody has to get to that state. So again, this is a gene that's being inserted into a virus capsid. So it's not a virus. It doesn't have all the pieces of a virus. It can't reproduce, but it is a way to get genes into cells. And in this case, it's an IV infusion. These are great slides because they're very graphic. The delivery mechanism has not been shown to cause any disease. Remember, it's been compromised and modified, so it's no longer an infectious agent. And what Astellas will be looking at is the changes in the heart as a consequence of receiving this genetic medicine. The protocol for the study itself will involve a screening. So people will have to come in and meet the criteria to be involved in the study. If you meet criteria, then you will be put into different cohorts. Again, we've talked about different dose groups. The first dose group will receive, as in a staggered way, doses of the gene therapy, and there'll be some time in between individual participants to make sure that it is safe and that everybody is doing well. Once we've gotten the first cohort done, as we've seen in the Lexeo trial, then we'll start recruiting for the next cohort, generally a higher dose than the first cohort. And just like any other gene therapy trial, there's a long follow-up period. So a year. I guess I forgot to say that, remember, this is a one-time administration. So unlike the other drugs that we've talked about, a gene therapy you get once, and then we wait and see how well that has, what benefit that has provided to you. So you'll come back frequently in the first year and then annually after that for about 5 years. So it is a pretty long commitment. This graphic tells you a little bit about what's going to happen during those trials. There is an inpatient or a hospitalization period just to receive the therapy itself. Again, it's new technology, really cutting edge. So safety, safety, safety. Then there are measurements that will be taken at different time points on both the heart and other aspects of the disease to see how well patients are doing. And that is frequent in the first year, and then it becomes just annually after that. As we talked about before, this study will be in adults. Again, that's a bit of a reflection of some conservatism in our regulatory agencies, something that Ron will continue to push on. But today, this will be an adult study of a certain weight and size and an echocardiogram, so imaging of the heart to show that you're a good candidate for improvement in that heart function were the gene therapy to work correctly. So there are some things that would eliminate you from participating as well, and they're listed here. One of the things that FARA has worked on for the last few years, and that you're starting to see the real fruits of that labor, is developing an understanding of the disease from a biological standpoint. biomarkers, things that are the biological aspects of the disease. So we have clinical assessments. How do you feel? How are you functioning? But we also can make decisions if we can look at the biology a little bit closer. And each of these programs is doing that. And here's an example where looking at changes in the heart, changes in blood markers, gives us a first look. Is this going in the right direction? And again, in this trial, you will be asked to give a sample of heart tissue, a biopsy. There are many sites, or at least several sites, through the US. So Brian, US only, correct?
Right, that's a QR code. It takes you to clinicaltrials.gov.
Okay. And you see sites here, all are enrolling right now?
We have our first site slated to open by the end of the month, and the remainder will follow them.
That's great. All right. So while I have you, Brian,
Sure.
can you tell us a little bit about, what you want us to understand about the Astellas program in particular?
Well, since you have the slide up, I think I'd like to first thank all the different sites for agreeing to participate in the study, and second, say that we are actively looking for participants who'd like to be part of this trial and who are interested in helping to advance therapeutics for FA and have access to this type of cutting edge therapy. So if you or somebody you know is interested, please reach out to either to Astellas or to one of these sites directly.
And can you just repeat the enrollment, the time frame for enrollment?
Right. So the enrollment period is there's a 60-day screening period that covers the time from your first visit at the site all the way up to treatment. Our first site is opening this month, and the subsequent sites will be following soon behind.
Okay. So before the end of the year, we
Yes.
definitely will see.
Yes.
There will be the opportunity to enroll.
Yes.
So I'm going to ask Brian hard questions. 'Cause
Okay.
I did this before and he did very well with it. If you have a therapy for a gene therapy for the heart, can you get a gene therapy for the neurological symptoms, the brain and spinal cord involvement in FA in the future?
So with current technology, that's not possible. The reason being is that all of the viral gene therapies that use a viral protein to encase and deliver the gene, all of those come from viruses, and our immune systems are very good at identifying viruses and mounting an immunologic response against them. So the expectation is that when a participant's body sees the virus that we're using to deliver the gene therapy, they will develop an immune response against that virus. The specific virus that's used would then not be a candidate to deliver a gene therapy again in the future. There are possible ways to overcome that immune response, but at the present time, the technology doesn't exist to deliver again with the same virus because of that response.
So if you were to receive or wanted to receive a gene therapy for other aspects of FA, it would actually have to be a completely different delivery system.
That would be my expectation
Yeah.
with the present technology.
Yeah, with the present technology. But remember, we didn't even have gene therapy a few years ago. So the technology is evolving. So you've got this first study, you're enrolling before the end of the year. Tell us a little bit more about the program looking forward from that first study.
Sure. Typically, results would come in from a phase 1 study. There would be analysis of those results, some discussion with regulators, with other experts in the field with the study participants and groups like FARA to decide how best to proceed. And then for a gene therapy candidate, there's typically a move directly from a phase 1 to a pivotal phase 3 trial. Those tend to come in a few different flavors. Sometimes the gene therapies are delivered to participants, and the comparison is made to natural history data. Sometimes the gene therapy can be compared to a placebo group. Sometimes it's compared to an active control. It really depends on whether there's a standard of care available and the type of therapy that's being used.
That's great. And we didn't have this question before, but, you know, if you can't answer it, I understand, but will people be allowed to be on Skyclarys and enroll in your study?
Yes, there's some restrictions based on things like oral contraceptives. This came up in Dr. Lynch's presentation earlier. If you are taking Skyclarys, you have to be on a highly effective form of birth control, just because we want to prevent any unintended pregnancy shortly after the gene therapy procedure. And the other thing I would mention is that we want to know that a person who's taking Skyclarys is stable on that drug before they would take gene therapy. So we're looking for people who either have decided they're not going to take Skyclarys at all, or who have decided they are going to take it and have been taking it for several months prior to gene therapy. And again, that's just so that we can distinguish any effect of the Skyclarys from a gene therapy.
That's
Thank you.
great. Thank you. That's really helpful. Do we have a few minutes to take questions? Great. Okay, so our panelists are in the order that they're listed here, sitting from Rusty to Brian. Questions from the audience.
We have an online question that we'll do first because it came in the beginning of the panel. So this is a question for Larimar and Barbara. Maybe you can also weigh in. Do we know anything about the safety or efficacy of OMAV plus the Larimar drug Nomla?
So I guess that's me, right?
I think it is, because I'm not sure I know the answer to that question.
So in our phase 1 study, any of our phase 1 studies, we didn't allow co-administration of omavaloxolone and nomlabofusp, which, by the way, you could just call it Nomla. That's fine. You don't have to worry about that funny letters at the end. But in our open label extension study, we are allowing the co-administration of omavaloxolone and nomlabofusp. And so far, in terms of our enrollment, it's about 50-50. We have about 50% of the population who's on OMAV and 50% of the population who is not. And so we're gathering co-administration safety data in that study.
Could you also explain the cocktail approach a little bit?
Sure. And the reason I get very excited about the diversity of the pipeline, the colors on the pipeline, the different mechanisms of action, that if you look at any other disease, and certainly a disease as complex as FA, it takes more than one drug. Even with gene therapy, depending on when it's given, there's probably an opportunity for improving quality of life with other therapeutics. So we expect that we need approval of several of these drugs in order to really be able to treat everybody at every stage of the disease. And that's our goal. Our goal is that everybody gets benefit, no matter where you are in your journey in FA. And that won't be possible with only a single therapeutic. So we're not out of business yet, Jen. There's still work to be done. But again, exciting that there's this diversity of approaches, because I think that's what we're going to need. Oh, Ron, don't ask anything hard.
You know what I'm gonna say
Okay.
too. But So I want to thank all 5 of you for excellent presentations and so instilling so much hope and confidence. You're helping us change our hope into confidence that we're going to get this one. Along those lines, I want to ask a question of our 3 gene therapists. We've all heard that you've begun your studies or will begin your studies with adults. And we know from Barbara why that is. I want to refer to the same July 16th event that Rusty and Barbara and Jen have all referred to today. That was the Pediatric Inclusion Roundtable that I remember seeing some of you there. And that whole day discussion on pediatric inclusion was kicked off after my opening remarks by the 3 medical product division or center leaders, Cedar, Sieber, and CDRH, all of whom spoke emotionally, almost as emotionally as I do, about the need to get kids involved in clinical research early so that we can begin to pivot from trying to protect them from clinical trials to trying to protect them with clinical trials so we can prevent their symptoms rather than trying to treat their symptoms later. Peter Marks, the center director for the Biologics Evaluation Research Center, the ones that review all gene therapy applications, was probably the leader of the pack in that regard, talking about how eager he is to use 2 things: accelerated approval for gene therapies and gene editing, and also to get kids involved in gene therapy clinical trials. Then the director of the newly established super office in CBER of therapeutic products, Nicole Verdun, spoke passionately about the fact that she is pivoting from trying to protect kids from, with trying to protect them with, and she said out loud, I no longer abide by the principle that we always must begin with adults. So you've established your protocol to begin with 18-year-olds and older. What I'd like to ask you is if you have initiated the conversation with the Center for Biologics Evaluation and Research, who have stated publicly their eagerness to get kids involved. Have you initiated that conversation so that you won't have to wait the 5 years to get regulatory approval or assent in adults to begin a pediatric study? Have you begun those conversations?
So I'm happy to share the same excited that you, Ron. Is our intent to include pediatrics in the next stage. Our trial is pretty much, the first one is pretty much fully enrolled. We're going to complete that probably in the next month. The next trial that is next year, that is the registration one, will include pediatrics.
So thank you for the question. And first I'll say that the Astellas team's keenly aware of the effects of FA on the brain, heart, and other organs in children. And we are keenly interested in addressing this age group. I think the best way to describe our stage is that we're having those discussions internally at Astellas right now, so that we can align on our own, how do I say it, rationale for how to address it best with the regulatory agencies. But our regulatory lead on this project was at that session and came back with a lot of interesting ideas, as did our patient advocacy lead for the project. So thank you for organizing it.
All right, I'm going to ask one more question from the online audience. And this one, Rusty, is for you. What does it mean for Larimar to be a part of the FDA START pilot program?
Oh, so thank you very much for asking that. So that was one of our nice surprises that we got this year was the FDA, and again, Peter Marks spearheading this as well, realized that for rare diseases, particularly neurodegenerative diseases, and Friedreich's ataxia is classified as a neurodegenerative disease, that there needs to be a faster way to get to ultimately approval. And the way to get into that fast lane is to increase the velocity of communication between the sponsor, the developer, and the FDA. And so Dr. Marks took a look at Operation Warp Speed that got us a vaccine for COVID in record time, right? So anybody on this panel or anybody in the audience in the drug industry would have said, we'll never get a vaccine in a year. And yet it was done. And it was done because of that rapid communication. And so the START program is this principle now applied to rare diseases. So Larimar put in our application in January, and we were lucky enough to be one of the few companies that were selected for this program. And we've now begun that rapid back and forth communication with FDA. So what would have taken us 75 to 90 days to get something of an answer, now we can get much more rapidly. And it's been able to accelerate our program. So it means a lot.
Well, I want to thank Barbara and our panelists for an excellent session. One final comment as we wrap up. So Barbara and I were presenting these pipeline panels and sharing with you a lot of the information shared by our panelists, by these companies. All of that is public information. We don't have any additional, we keep saying we because all of these folks are our partners, but we don't have any information that's not publicly available. And we also want to stress, our partners and FARA, that it's always important to talk to your physician whenever you're considering any clinical trial. A conversation with a trusted healthcare provider is critically important. Everybody's situation's a little different, and we just want to remind you of that.